All right, everyone. If you haven't done so already, please take a look at the welcome video for this class. And each welcome video, if you're watching this in the future, each welcome video will be pertaining to the specific semester that we are running this course. And so, welcome. Anyways, so if you haven't taken a look at my welcome video, just a recap of how this class is designed. I posted all of my lecture videos, at least at the time of this recording. I have most of the lecture videos already recorded and ready to go. And so hopefully you guys are able to kind of just take a look at it. Now, how I want you guys to kind of approach this class is to use these lecture videos for one of several reasons. Number one, you forgot to come to class that specific day and you need to catch up. These are the lecture videos for you. They're going to go over everything that we are going to be going over in class. Number two, I would like you guys to review these lecture videos ahead of time. And the reason being is because classroom time, at least in pertaining to in-class sessions, we are going to be mainly going to touch up on the main important bullet points within the chapter itself, as well as going over practice problems. I do want to have that time for you guys where you guys can ask me questions and then we can work on several of the book problems together during class time. So it's a way to kind of flip your studying. You study during class while we are in class together, while I'm there to help you guys. And then at your own pace, you can watch these lecture videos. Now, these lecture videos will be divided into several pieces. I recommend doing 20 to 30 minutes a day before the next class meeting. Some chapters will take a whole week to go over. Some chapters will be done in about a day or two. But I predict that most of these uh, videos will be roughly between an hour, hour and a half long. And so, without further ado, let's get started. We are in chapter one. This is the beginning of the whole series. There's roughly around 16 to 20 chapters that we're going to be getting through this semester, about one chapter a week. And fortunately for the organization of the textbook, the first several chapters should be a big review for you guys. So if you guys haven't done so already or have done so, and you just recently took the last course of Gen Bio, Gen Chem, or even organic chemistry, the first 2 to 3 to 4 chapters are going to be a very big review. And what I mean big, I mean it's like really big review. We will be going over the base fundamentals of what you will need to know for this upper division course, and then we're going to go from there. So let's get in, let's get started, and we're going to get started. Just jumping on in, so you guys can see I'm really doing this improv, and so hopefully you guys are going to enjoy this. Anyways, chapter one: cells, genomes, and the diversity of life. Now, if you guys remember from your Campbell textbook, or if you guys still use the Campbell textbook or your Gen Bio textbook, the first chapter is always about the organization or the description of what makes a cell a cell. Now, in this specific chapter one, we're going to expand on that. Not only do you will need to know the base fundamental of what life is characterized by, but now we're going to get more specifically into what makes a cell living versus non-living. And this is going to be divided into several pieces, and so we're going to get through it right now. So for the majority of the organization of these PowerPoints, the first several slides are going to pretty much list out what you should know concept-wise individually. So there are 4 pieces to this specific chapter. The first one is the universal features of life on Earth against what all life would be characterized with. The second piece is genome diversification and the tree of life, how the genetic makeup of that specific cell is shared throughout the living organism tree, biome tree. The third one is eukaryotes and the origin of eukaryotic cells. And again, it's a review of what was the initial life when life first started on this planet as prokaryotes and archaic cells into eukaryotic cells. And then what do we use specifically in science today as the model organism, whether it's bacteria, a single-celled organism, a eukaryotic organism, plants, etc. And so we're going to go through each of these 4. This will be a really good summary of what you should be expecting to know within the textbook itself. And let's get started. So the first, I'm gonna pre-warn you guys right now. The first several PowerPoints is gonna be a huge list. Again, it's expected that you guys know this already since you guys are in upper division bio, but we're going to go through it. I'm going to explain a good chunk of the slides, but I'm not going to go into specific details as most of this is review. Okay. Now, first things first, we're going to go over the universal features of life on Earth, and you're going to need to know this. All cells, and here's the several bullet points. And most of these will be probably common knowledge for you guys at this point, but all cells store their hereditary information in a double-stranded DNA, some form of a double-stranded DNA. So again, DNA is that blueprint of all living organism. Whether you're looking at the homeobox or you're looking at the whole genome itself, there are going to be specific pieces that are highly, highly conserved throughout the whole genome. From organism to organism, there will be some repeating things that we will be highlighting later on this semester. Replication of the hereditary information. Again, we got to replicate our DNA either from a double-stranded to another double-strand or from double-strand DNA to a single-strand mRNA or RNA, etc. Transcription portions of their DNA into RNA molecules. Again, that's the repeat of the second bullet point, or they go into both. They use proteins as catalysts. Now, specifically, you guys should know this already, especially when you guys already understand what enzymes are. You know that they lower the activation energy, they act as a catalyst. But remember, not all proteins are enzymes. This is a big thing that my students always get confused. Not all proteins are enzymes, but all enzymes are proteins. Now, this bullet point is saying proteins in general. So just be very careful with this because there are certain proteins that are not enzymes that will also aid in a catalytic reaction within the cell. Translation of RNA into protein, again, same. You guys know the central dogma, DNA to RNA to protein, and function as biochemical factories. Now, this is kind of, if you guys want to connect your general courses together, this is what we taught you guys in the second half of general chemistry, where we're looking at kinetics, enzyme kinetics of a system. Now, we are biochemical, so living chemical systems that run chemical reactions. So we're going to be kind of reviewing a lot of this, especially when you're getting into enzymatic reactions. Okay. Now, each protein is encoded by a specific gene. As always, and life requires a continual input of free energy, meaning you got to find energy to produce energy. It's an energy conversion. This goes back to your gen physics, if you guys have taken gen physics already. Okay. So, again, this is a whole list. We're just going to go through it. All cells are enclosed in a plasma membrane. So yes, even plant cells, and a lot of my students always forget that plant cells, not only do they have a cellular wall, but they also have a plasma membrane underneath that cellular wall. Just like with any type of armor, if you're going to wear a piece of armor, you're going to have a squishy underside versus the hard outer plated armor. So cells are the exact same way. Plant cells or bacterial cells that also have cellular walls also have a plasma membrane underneath that armor. So don't forget that. Across which nutrients and waste material must pass through. Again, we got to expel waste products. Cells operate at a microscopic scale dominated by random thermal motion. Again, this goes back to 111B and a little bit of 111A, where we do talk about the movement through diffusion, osmosis, and facilitated diffusion. It isn't like a thought process like, hey, I want to move a ping pong ball from one room to the other. It just happens through a thermal process. Thermal meaning energy, heat energy. So just FY. And living cells can exist with as little as 500 genes. So you can have a cell that exists with only 500 genes and it will be a viable living organism. Okay. So again, what does this mean? Well, if we look at the origins of life, and if you look at the development of an embryo, we all start off as a single-celled uh, depending whether you're a bacteria or a eukaryotic cell. But for a eukaryotic cell, we are initially going to start off as two separate cells, right? You're going to have the sperm, you're going to have the egg until they form a fertilized cell called a zygote. I'm using a lot of terminologies right now as a review. And we can see on this image right here, in this slide, you can see the fertilized eggs at their beginning stages of development. Now, you're still at the single cell stage. We haven't developed into the morula or the blastula. We haven't developed further, but you can see the similarities between the three, whether it is a sea urchin or a field mice or even a plant. I think I want to say that's a kelp plant, but I'm not too sure. Might say it on the side, but I don't know. But we can see that in all three cells, you you see the similarities of the egg cell fertilized. You can see maybe some of the DNA molecules in there, maybe. But one thing that you do want to take a look at is the size ratio of each of the cell. The sea urchin's egg, fertilized egg is around 100 microns big with a eukaryotic mice as well as a plant, you can see that they're roughly between 50 to maybe 100 microns big, at least in terms of the scaling. And so that's something that you want to also take in mind. This book is heavily uh, trying to depict the visualization of the sizes of organisms, whether you're looking at a bacterial cell versus a eukaryotic cell, you're going to see that size plays a role, whether it's going to be a couple hundred microns to something even bigger than a couple hundred microns. Just keep in mind of that. All right. Now, another thing that all cells contain, again, all cells store their hereditary information in a form of double-stranded DNA molecule, and they need to find a way to replicate that. We're going to go through this list, okay? And so through different types of process, here is the base blueprint of what you should already understand. So I'm going to start introducing another topic right now. This we cover this in chapter 2, but since you guys have already have a background in biology, you'll know you have monomers and you have polymers. You know that the four macromolecules of life is proteins, carbohydrates, lipids, as well as nucleic acids. So, DNA. DNA falls under the category of nucleic acids or nucleotides, nucleosides, depending if you have a phosphate group attached. Now, in this depiction, in this figure right here, we are showing you guys the process of building a nucleic acid chain. Whether it's a single-stranded or a double-stranded, it will follow the same pattern. First, you need to create the monomeric pieces, your nucleotide. Now, the difference between a nucleoside and a nucleotide is whether or not the phosphate group is attached to the sugar backbone as well as the nitrate base. Okay, so in figure A right here, and let me get my pen out so that you guys can follow along. In figure A, we're looking at this figure right here. You can see that you're going to have a sugar phosphate. So the phosphate is already present and it's going to bind to one of the four nitrate bases. So you have your purines and your pyrimidines. You have your adenine, guanine, thymine, and cytosine. I can't remember the full names of them off the top of my head, but just know that A binds with T, okay? Adenine binds with thymine, and guanine binds with cytosine. Now, also keep in mind that how they are linked together is through the process of hydrogen bonds. So I am going to introduce a lot of things right now because this is going to be talked about in chapter 2. And if I talk about it now, we can kind of breeze through chapter 2. But just know that the more cysteine and guanine binding that you guys have, the more thermal I wouldn't say thermically. I would say that the more stable the DNA structure becomes at higher temperatures. Again, it just means that you have more hydrogen bonds, more magnetic bonds, so to speak, to keep the DNA bounded in the double helix form. And you need more energy to break hydrogen bonds away if you have more of them. So again, we're trying to piece the concepts here. But just know that as we are building our monomers, our the nucleotide bases, and connecting them together, we are going to create a single-stranded DNA. And then from a single-stranded DNA, we're going to use an enzyme to lower the amount of energy required to build the second strand because now we have a template. We have a template strand, okay? Template strand. And now, as the polymerase comes in and starts creating the secondary chain or your complementary strand, then it will form a double-stranded DNA. And then from there, we want to kind of condense the DNA down even further. So we're going to have a double helix formation, and the sugar phosphate backbones are going to have a positive or negative charge. Sorry, phosphate has a negative charge, and you're going to have a dipole-dipole situation or polarization situation in the DNA itself while it's forming a helical shape. Okay. So again, template strand versus the complementary strand or new strand, as your textbook says. We are a semi-conservative model. Again, understand the difference between what a conservative, fully conservative, semi-conservative, and non-conservative replication is. We as a eukaryotic cell, we undergo a semi-conservative replication pattern. Okay? All right. And so again, with that, now we understand a little bit, kind of review over how DNA is made, kind of getting familiar with the vocabulary again. Know that this is the key in all biology textbook, your central dogma. Okay. This is your central dogma. And this is the path that all cellular life will take. You go from a DNA strand, your blueprint, you're going to make different types of RNAs. You can have your mRNA, your ssRNA, your tRNAs, your rRNAs, whatever type of RNAs you're going to have. You're going to have it because then it will create specific proteins, which will aid in your biochemical reactions found within the cell. Okay. Again, when you're going from DNA to RNA, this is a transcription process. Transcription is DNA to RNA, and then RNA to protein is your translation. Okay. I know it says it in the slide or on the image on the slide right here, but just to really emphasize the importance of understanding what each step does, make sure you know these terminologies. Okay. And again, if you're going to go for DNA to DNA, this happens during the replication process, especially when the cell's going to undergo division, okay, during the S phase of a cellular's life cycle. Okay. And it also uses protein. Obviously, we need protein. Protein is a vital source, a vital molecule that aids in the biochemical process. Now, I use a term for my 212 labs when I'm teaching about enzymes. And I'm going to share it with you guys. Some of you guys might have had me for 212 already. Some of you might not. And so for those who don't remember, enzymes and to an extent catalytic proteins are tools that our cells use. Okay. They are just that. They are tools. Now, imagine what a tool does in your life. Say that you need to hammer in some nails on your wall to hang picture frames. Now, if you don't have the proper tool, you might find that you're going to spend a little bit more energy. Now, imagine you don't have a hammer. I gave you a box of nails, and I asked you guys to nail 5 picture frames on the wall. You don't have any tools besides the nail and your hand. Now, if you try to nail the nails onto the wall using your bare hand, you're going to find you're going to have to use a lot of energy to slam the nail into the wall. And most of the time, you're not going to have that perfect placement of the nail. So imagine how much energy that took. Now, say that after this project is done, I want you to go to another wall and hang up 5 more pictures. But this time I gave you a hammer. Now, immediately you'll understand that, hey, I know how to use hammer, and you're going to get it done maybe within 5 minutes. Versus prior to without the hammer, it took you a much longer time, maybe 20 minutes. And it hurt your hand versus with the hammer, it didn't hurt your hand as much. So the tool is a way to kind of speed up a process that you want to get done. In this case, a chemical reaction. In our example, hammering the nails into the wall. But it's also a way that to kind of visualize what enzymes and what catalytic protein does within our cell. Now, note that the hammer will last as long as the hammer will last, right? You don't want to just use a hammer once, toss it out, and then bring it back, buy another one, and use it again. It's just very inefficient to that. You're going to reuse the hammer until it breaks. Our cells does the exact same thing. It's going to reuse these enzymes or catalytic proteins until it no longer functions properly. Then it gets recycled by through digestive process to peroxisomes or through digestive proteins. It gets recycled and then remade into another catalytic protein. So again, I'm just reviewing with you guys. You guys should know this already, but this is a way to kind of visualize how efficient our cells and to an extent other cells are when it comes to biological reaction. And again, we use proteins, aka enzymes, as catalysts. Okay. So again, list, I already talked about this, biochemical factories, continued input of energy, and all cells are enclosed in plasma membrane across which nutrients and waste material must pass. So for our particular cell, we form a lipid bilayer where we have a layer of squishiness, aka fats, phospholipids. And you can see in this imagery right here that you have a lipid, a phospholipid more specifically. Now, to break the lipid back into its monomeric form, and this is just a review. Remember, a lipid is comprised of 2 specific monomers. Okay. You have the first monomer, which is your glycerol cap. Now, glycerol is a 3 carbon molecule, okay, with OHs and hydrogens, hydrocarbons, whatever. But a 3 carbon backbone molecule. And attached to each of those carbons, especially when we're making different types of lipids, whether it's phospholipids, triglycerides, etc., you need to have your fatty acids attached to them. Now, what are these fatty acids? They are your omega-6, your omega-9s, your omega-12s, your omega-15s, your oleic, linoleic. I forgot the other names of the fatty acids, but these are those fatty acids where we are going to bind them onto each of the carbon molecules through a process of dehydration. Okay. We're going to bind them and we are going to either attach a phosphate group or fatty acid tails on them. And depending on what you have, for this case, this right here is 2 fatty acid tails. So this is a fatty acid tail 1, fatty acid tail 2 with a phosphate group. This is what we call a phospholipid, okay, which has a hydrophilic portion and a hydrophobic portion. Hydrophilic due to its polarity of the makeup of the chemical makeup. Okay, your hydrocarbons right here, your fatty acid tails are all carbons to hydrogen. Doesn't have any polar or huge polar differences, polarity differences between the carbon and the hydrogen atom. Versus a phosphate group, which has 4 oxygens bounded to a carbon, the oxygen carbon and the phosphate and oxygen will create a polar behavior where it likes to interact with polar molecules. So again, depending on what you have, if you have a layer of oil on top of water, you're going to see a separation where the phospholipid hydrophilic head is going to be interacting with the water and vice versa. The fatty acids will interact with the tails. Now, when we have a cell that's formed like this, okay, again, it's a double layer. So you have one layer on the outside, one layer on the inside, but sometimes you also have a single phospholipid layer, which we call a micelle, that transports specific types of molecules within the cells itself. So you can either have as a vesicle transporting from one part of the cell to the other, Golgi to the outer membrane, etc. But just keep in mind these fundamental shapes. Okay. Now, cells operate at a microscopic scale dominated by random thermal motion. Again, you have plasma membranes, you have actin filaments or the cytoskeleton underneath, and it's going to be moving back and forth. Now, I think this image is misplaced. It should be in relation to the last slide. But just know that when you're sitting in a room, you can feel thermal energy, especially if it's a hot summer day like it is today. You can feel the heat build up in the room. Now, why is it that we feel that heat? Is due to the fact that molecules are hitting our thermal receptors found on our skin. And depending on how often a molecule hits that, we can register that as being hot versus cold. And again, this is exactly how a regular room thermometer works. It measures how often a molecule is hitting the the little red bead, if you're looking at a regular thermometer, thermal thermometer, you're gonna look at that, and how often that molecule hits that bead will show in terms of the temperature gauge, how hot or how cold it is in the room. So as long as you understand that concept, you should be able to understand that molecules within a cell is moving because of this thermal motion. Now, again, thermal is just a fancy way of saying heat energy, but again, anything that moves needs to have energy. So heat is a form of energy that a molecule can absorb and then use that for a kinetic movement. And here again is an example of a cell that can exist with as little as 500 genes. Now, your book goes specifically into what type of cell this is. I recommend reading the textbook, but just know that 500 is the little magic number that is needed for a living cell to occur. Okay. So at this point, we are done with the first of the four parts. If you need to, take a break now. I highly encourage you guys to take a break, come back later, right? We talked about 30, 40, maybe 20 to 30 minutes at this point. And don't try to watch this all at once. Really, just take a breather, pause the video, go to the restroom, go outside, and then come back later in the evening or later in the morning to watch the second part. Okay. That's my recommendation. But if you want to keep going, we're going to keep going. Hmm. Okay. So we found all the characteristics that life shares more specifically than what your Gen Bio book goes through. And now we are in our second part. Genome diversification and the tree of life. How do all organisms relate to each other as well as not, right? How is everything classified and organized? And so here is a list again, is a bullet point list, and we'll go through each of them one by one. So, tree of life, you already know this, is divided into three major domains. Okay, so you have domain and then the extra classification. We're not going to go through that. Again, you don't need to know the genus, the species, and everything yet. But just know you have your eukaryotic cells, your bacterial cells, your prokaryotic, and your archaic cells, your archaea. Now, eukaryotes make up the domain of life that we are most familiar with. Again, we are eukaryotic cells. And on that basis, bacteria are the most diverse group of organisms. They're part of the largest in terms of different types of species. Okay. Archaic cells are probably the not most understood, but they're the most mysterious domain of life. We don't really talk about them too much. There are specific fields where, our archaic biologists are studying right now. But it's something that we're kind of like indecisive about. Not indecisive. It's just a huge mystery at this point. Now, our organisms occupy most of our planets, and cells can be powered by a wide variety of free energy sources. Again, that's a repeat from the prior section. And some cells fix nitrogen and carbon dioxide for other cells. Now, one thing that we need to really, really kind of understand, and I'm hoping that you guys have a background of ecology at this point, but everything is related to each other somehow. Bacteria is related to eukaryotic cells. Plant cells are related somehow in the whole web of life. We need certain organisms to really run the whole entire ecosystem in the biome, etc. And so, ju just understand that bacterial cells, yes, some of them are very detriment to our livelihood, especially if you have one of those mutated bacterial cells like MRSA, uh, where they're resistant to antibiotics and they start eating human cells. That's something that we don't typically want to happen. But there are very important, especially decomposing cells that are required for our ecosystem to thrive. Because without them, especially nitrogen fixing bacteria and or eukaryotic organism, without those cells, we really would run out of nitrogen um, from the environment. I mean, our agriculture is heavily, heavily dependent on the amount of nitrates found within the soil. And without them, a lot of the food being produced today, agricultural food produced today, is very undernourished. So, that just a little 2 cents on how our world is connected. Genomes diversify over evolutionary time. New genes are generated from pre-existing genes. Again, we need templates. Gene duplication give rises to families related genes within a single genome. So that just means that a specific family within a DNA sequence. So you can have like your ribosomal families, right? They might have multiple genes that creates multiple different types of ribosome, but they still fall under the classes of ribosome or vesicles or, etc. Okay. The function of genes can often be deduced from its nucleotide sequence. Again, we've done enormous amount of studies studying the DNA sequence and be able to figure out, especially with AI intervention now, be able to piece together how a protein would look or how a specific gene being converted into a protein would look and how that would interact with the rest of the body. And more than 200 gene families are common in all three domains of life. Again, this is a reoccurring thing. We came from a very specific genetic makeup, a common ancestor, if you will, depending on how far you want to go back. But there are multiple repeats of the same type of genes and gene families. So here is just a visualization of what you can find on the internet. The different classes of bacteria, the fungi, the archaic cells, the eukaryotic cells. And you can see that our little biome right here of eukaryotes, we only make up this much genetic diversity. Bacteria, again, makes up all of this. This is your salmonella, your E. coli, your what other cells you want to call? It just makes up everything. Psyllium, not psyllium cells. What is it? Cyanobacteria. All these different types of single cell bacteria that thrive in our world. And again, archaic cells, again, is this little region right here, which is not fully understood as of right now. Okay. Now, eukaryotic cells make up the domain of life and bacterial cell. Let me go back. Bacteria are the most diverse. We see that through that tree. But here is an image of different types of bacterial cell. Now you can see that some bacterial cells are going to be spherical in shape, others are going to be rod-like, others are going to have a tail attached to a ovular shape cell. Some will have multiple different types of shape inside of them. But you should be able to see, compare from that first image that we showed you with the eggs. Remember the sea urchin, the the plant as well as the was it a bird or chicken? I forgot what the other animal was. Remember, they were 10 microns big, 10 micrometers big. These bacteria cells are one-fifth the size or less from those big scalings. So these are incredibly, incredibly, incredibly small. And being able to fight off these type of bacterial cells is somewhat easy for eukaryotic cells until the point where they are resistant to the different types of antibodies that we have or medication that we take. That's when it starts getting into a very dicey area. Okay. So here you can see very similar makeup. They do not have a nucleus. Again, they're too small to contain any type of organelles. So all genetic material is floating within the cytoplasm itself. You have your DNA in there, you have your ribosomes, you have your RNA, you have your specific structures like the flagellum, the tail of the bacterial, the cell wall is needed to protect the bacteria, etc. So, and you can see right here again, the cell wall, just to reiterate, the cell wall is this yellow region right here. It's the armor of the cell, and right underneath it, you have your plasma membrane right underneath it. Okay. Here's another image of different types of bacteria. We can see the cellular makeup, the cell wall. You can see the little DNA strands in here. Maybe the little specks right here are your ribosomes right here. Again, this is just to show you guys the scaling difference between 10 microns versus 1 micron. Okay. A different type of image again, different type of bacteria. And archaea is the most mysterious of all. Okay, so genome diversification. Here is an image right here that shows. I forgot what it shows, to be honest. I can't remember what this image is, so I'm going to skip it, but please refer back to your book. GTCs. I want to say this has something to do with genome thermal temperatures or something. I don't remember. So we're gonna skip it. Anyways, refer to your textbooks, everyone. All right. So cells can be powered by a wide variety of free energy sources. What does that mean? That again, it's energy sources that are found outside of the within the environment. So you either have solar energy, you have chemical energy, or you have heat energy, thermal energy. And in the deep sea uh, ecosystem, you have geothermal energy. So the thermal energy directly coming from the heat of the earth. And so here's an example of different types of organisms that thrive on geothermic energy. We can see that, depending on the location and how but further beneath the seafloor is, you can see that depending on the activity level or I want to say the circumstance of the shape of the crust at that specific time, you will have the ability to shoot through hot magma slash lava. Okay. Now, a lot of that's under pressure, high immense amounts of pressure from the core of the earth that brings out that temperature. But we can see that it's around 350 degrees Celsius. And once it shoots out, it cools down drastically. However, because the seafloor is in this region and you can have water going through in and out, depending, uh, it's going to warm up the surrounding area. And there are organisms that have been able to capture that thermal energy and use the extra thermal energy with the movement of their internal molecules to be able to run a biochemical reaction. And so right here, geochemical energy and inorganic raw material, bacteria and archaic cells digest those materials or utilizes those materials somehow, and then expels out organic material that other organisms can ingest and use as energy. Now, another example would be solar energy, and we're very familiar with solar energy because we have photosynthetic organisms. So whether it's an algae, eukaryotic plant, or even photosynthetic bacteria, where we're using solar radiation, absorbing that, converting that into a usable form of energy through the conversion of carbon dioxide into sugars, and then from sugars into cellular energy known as ATP. So these are just many, many different types of ways that we can use these processes. Now, this image right here is another image where we can see the very, very, very similarities between humans and different types of single cellular organism. Right here, you have the methanococcus and the E. coli, and we can see where there's a red line is where the DNA did not change or mutate over the millennia. Okay, we can see these are highly conserved regions of the DNA. But with small mutations here and there, this will add a change in the protein structure potentially. And then through the change in the protein structure, your phenotype, your physical trait also gets altered. And this is how mutation occurs over generations into also evolutions from one organism to the next. This is again reiterating the state, restating. I'm losing words here, but this builds into the idea that the genes are generated from past genes. Okay. Okay. Again, genes give rise to duplication with families related genes within a single genome. 283 genes in a family with 38 to 77 gene members, etc. This just states that you can have multiple genes within a single genetic region or a single genome, and it will give rise to multiple classes of them. Okay. So right here, again, ancestral organism, you have gene, a specific gene, and speciation occurs where they're either isolated habitually, geographically, etc. And then you form two separate genes. You have gene A versus gene B, and these are known to be orthologs with each other because ortho means one or the other or diverging next to each other. Okay, but they're specific to those specific organisms. Now, this in B right here, you have that same gene, okay, but through duplication of the gene within that same organism, you can have both gene 1 and gene 2 at the same time. Now, these are known to be paralogs, and they could be used at the exact same time. They might have the exact same function, or it might be under specific circumstances that gene 1 gets turned on and gene 2 is stays off, or vice versa. So, but if you can go between the two different types of genes within the same organism, that's a paralog. If species A has gene A only and species B has gene B, that is what we call an ortholog. Okay. Again, same thing, but we can see the process of analyzing the DNA, and this is just the same extent of what we do for not gene therapy. What is it called? When we're looking at your genetic makeup, and we are researching your own DNA. So you submit your DNA to Ancestry.com or 23andMe or whatever other DNA thing. We're gathering that intel, and then we're able to analyze your genetic makeup and see what health issues down the road we can predict will occur based on your genetic makeup. Again, we're able to classify all that right now, and this is why this is a huge issue within the medical and research community, where how far does it go into the privacy of your genetic makeup? Uh, do you want that information to be taken place in the pharmaceutical route where they can develop medicine for you or not? That's a huge another topic that we're not going to get into. So, but just know that this is essentially what is being shown here, where translation, information process, we know that there's 63 um, different types of genes that undergo translation or is involved with translation and etc. Okay. All right, eukaryotic cells and the origin of eukaryotic cells. We are, what is it? we are done with the second part. So I'm actually going to stop this video here. This is going to be a two-part video, so I'm hoping that this was enough to kind of get you guys started. But we're going to stop right here on the third part because we're going to go from the third part to the fourth part in a second video. But I'm hoping that you guys will take this time to watch this first video and just review over it. Again, this is all review right now. We're just kind of bullet pointing over it. We're kind of understanding how the pieces are fitting together again. And hopefully, as we go through the semester, you guys will be able to build on this foundational understanding into more advanced understanding. So, until next time, everyone, hope that you guys enjoyed this first video and see you guys in the second video. And don't forget, when you are coming to class, please come with questions. I'm hoping that you guys are watching this ahead of time, but please come with questions if you have any questions and be prepared to work together on some practice questions during class time. Okay. All right. See you guys in class. Take care, everybody. Okay, so I'm thinking the recording is going on right now. Usually it would say it's recording, but I'm hoping it's recording. Anyways, welcome back. This is day two or even part two of the chapter one lecture series. Now, let me give you a moment right here to kind of reset everything. Okay. And so, what do we just well, we talked about in the last episode was the pretty much the more detailed characteristic of what all living organisms shared, as well as the genomic diversity and close relatedness of how it branched off from each other. I know that didn't really make sense too much, but just know that all genes are derived from a common genetic template where over the millennia or over the generations, mutations occur, which leads to different types of speciation, different types of mutations, and phenotype and genotype traits, genotypic traits. And so from there, we are going to continue on to really defining what eukaryotes are and the origins of eukaryotic cells. And so, just really quickly, we're going to read through this list and then we're going to go through some examples. So, eukaryotic cells contain a variety of organelles. Again, we're going to treat eukaryotic cells like how we treat ourselves. They will have their own specific organs within the cell, hence, we call them organelles. And each of those organelles will have a very unique role within the cell uh I don't want to say biome, but the cellular system. Okay. Uh Again, endosymbiotic relationship with the mitochondria and the chloroplast, depending on which organism you are looking at, are pretty much ancient bacteria or archaic cells that have pretty much been engulfed with a eukaryotic cell and have a symbiotic relationship. Now, they're going to play a very vital role in our ability to produce ATP and or for plants or some autotrophs role to produce sugars. Okay, chloroplast evolves again in this symbiotic relationship. Eukaryotics have hybrid genomes. Again, we have two separate types of DNA. We have our own eukaryotic DNA as well as the mitochondrial DNA that gets passed on from your mother's mitochondrial DNA. So again, all mitochondrial DNA are derived from the mom, as the egg cell contains all the essence of what the offspring organism will have. The sperm cell only really gives off the chromosomal DNA from the father side, but that's essentially um, it. Eukaryotic genomes are huge. Again, we are complex creatures, depending on the eukaryotic organism, but they are much, 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 much larger than the prokaryotic and the archaic cells. And eukaryotic genomes are rich in regulatory DNA. Again, we need a lot of regulation. We don't want the genes to turn on all the time. We only want them to turn on when we need it to turn on. So we need to have a lot of coding in our DNA to regulate all of that. And so here we go. We're just going to go through again the examples within the PowerPoint here. And let me actually get my pen out just in case we need to write some things. And so here we go. Eukaryotic genomes define programs of multicellular development. Again, depending on what type of eukaryotic cell you're looking at, most eukaryotic cells are multicellular organisms. And so we do need programming for that. And many eukaryotic live as solitary cells. Again, it depends on what type of eukaryotes you're looking at. If you're looking at a yeast cell, that is a eukaryotic cell, a single-celled eukaryotic cell, or amoeba, those are all single-cellular organisms. And so, will amoeba fall under it? I can't remember. Anyways, so organelles. We all understand the properties of what each individual organelle does. The Golgi apparatus is pretty much the mailing center. The endoplasmic reticulum is responsible for synthesizing protein and some components of the cellular membrane. The nucleus houses the DNA as well as a lot of other regulatory genetic material, and et cetera, et cetera. Now, we're not going to go everything in specific detail in this chapter one, but we will delve into it much deeper as we go into later chapters, especially when we're talking about cellular signaling. Okay. Against cell surface protrusion, you have a dividing bacterium right here, plasma membrane. We know the process of endocytosis and exocytosis. So endocytosis is the engulfing of material, either it's a bacterial cell, food source, etc. But we're going to engulf them within and then digest them using the correct tools that our cells have, so peroxisomes and etc. Okay, we can see that this is an imagery of a white blood cell, which is engulfing and destroying an invading bacterial cell within the body. And so that's what we can see right there. Import by endocytosis, again, and you're going to have endosomes, you're going to have plasma membrane, you're going to have vesicle formation, micelle formation, etc. And you're going to be using a lot of the Golgi apparatus. Again, Golgi apparatus is the mailing center, the packaging center that is going to transport things from one organelle, either to the cell membrane or to other pieces of the other organelles of the cell. Okay, and export using exocytosis. So I teach a terminology class at another institution, and whenever we come across scientific terms, use the scientific terms to break down the scientific terms to kind of have a base understanding of the terminology itself. Now, most of you guys will already know what these terminologies are because you're familiar with them, but for those who are kind of still struggling with the various amounts of biological terms, this is a shortcut that I found to be very helpful, especially during my undergrad years a couple decades ago. And I still remember it today. And so when we're looking at endo versus exocytosis or just any type of terminology in general, okay, break the words down. Really break them down. Okay. We know and we've recognized the pieces of the words endo, endo and exo. We kind of remember what those are. Endotherms, exotherms, that kind of really just remembers, reminds us. And so whenever you see the word endo, you're going to think of inside, indo, indoors, inside. This represents going within, okay, inside. Cyto, we recognize that term, cytoplasm, cytocell. Okay, this is a cell. Okay, so when we're looking at endocytosis, and is a suffix for pertaining to. Okay, this is a process that pertains to going within a cell. So endocytosis. Again, I'm using just the pieces of the words that I recognize to kind of build that definition. And most of the time, this would be a good avenue to kind of shortcut the memorization of terminologies. Now, once you get into further into, say that you want to go into the medical profession, this is going to be very, very helpful because, there's a lot of medical terminology that is based on Latin roots, prefix, and suffixes. biology is the same thing. We base a lot of our scientific words on Latin. And so the more that you get familiar with the pieces of words, the more likely you're able to kind of define what these words are, especially if you're planning to take your MCAT later on. This is a really, really great skill to kind of start developing while you're studying for those pieces. Anyways, going on. So again, these are just pieces of the cell. You have your mitochondrion, you have your peroxisome, you have your cytoplasm or cytosol, endoplasmic reticulum, endosomes, etc., etc. But these are all the organelles and the pieces that pertains to a cell, and they're all interconnected in some fashion. Now, as we get closer to the process of cell signaling, you might have already studied that through biochemistry if you're taking 441. But these things are going to get more and more prevalent uh, as we get later on to the chapters. Okay. So eukaryotic cells and the origin of the eukaryotic cell. Mitochondria evolved from a symbiotic bacterium captured by an ancient archaeon. So all this is really saying is that we have a symbiotic organelle called the mitochondria that is not developed through our own genetic material. They have their own DNA. And so when we are looking at maternity tests or paternity tests and say that you're trying to figure out who your mom and dad is, mom is going to be much easier because we can match mom's mitochondrial DNA to your own mitochondrial DNA. And because of this, um, it's going to be much easier to figure that out. Now, the paternity test, again, you're going to go through the chromosomal DNA, and that's going to be a little bit varied. Yes, you're going to be able to have a good match and find your relatives through the chromosomal DNA or the main DNA. But this, again, this slide is just really saying that we are in symbiotic relationship with the mitochondria. And so we can see you have the archaean cell body, you have the archaean cell protrusion, and then the ecto symbionts or symbionts. Okay, so this is just an imagery of different types of archaic cells, and it's interacting with some symbiotic organelles here. Okay. So right here, bacterial ectosymbiotic, you have archaean DNA, you have bacterial DNA right here. And then what's going to happen is that it's going to have a process of endocytosis. So the bacteria is going to get engulfed within the archaic cell. And through that, you're going to create a nice little barrier. Now, because this is a symbiotic relationship, we don't want any type of, how do I say it? Any type of antibodies to attack it or any type of peroxisomes to attack it. So there's going to be some sort of protection with that bacterial cellular wall or cellular membrane. But right here, we can see that as it's getting engulfed, and let me draw the picture right here, we can see that as it's getting engulfed right here, we're going to have some expansion occurring so that it starts floating around in the archaic cytosol. And through there, we can see it's forming a nuclear envelope around. So enclosure of ectosymbiotic by archaeal membrane fusion. Again, we're trying to fuse and join it together. Escape of endosymbiont into a cytosol and formation of new intracellular compartments. So again, we can see right here, there is a little membrane, or not a membrane, but just a little area gap before the fusion occurs. And once it's fully engulfed within, certain chemical reactions occur, certain relationships occur where the cellular membrane and the cellular wall, the cytosol, the cellular wall or membrane, interacts with each other, recreating some sort of relationship, either a road with into and out of the cell, or some sort of intracellular structure. So, if you guys take a look at our mitochondrial cell, and just to kind of expand on that concept, our mitochondrial cell, we have to be able to feed it glucose, right? Through cellular respiration. And glucose can only enter that cell with the help of an acetylcholine or acetyl-CoA attached to the pyruvate. And so there's going to be some key criteria that will make things be able to go in and out of the cell. And this is what the slide is essentially trying to demonstrate. Again, we're not going to go into this right now. We'll go into it later on. Chloroplast is the exact same concept. It's going to get endocytosed into a plant cell or any type of autotroph cell, and the chloroplast will remain there producing some amounts of energy, but mainly the process is to convert sunlight energy or solar energy into a usable cellular storage form of cellular storage or cellular energy. Okay. Again, eukaryotes have hybrid genomes. We can see that a lot of the genomes are going to be shared between the three, as well as we contain prokaryotic, archaic genomes within ourselves or within the mitochondria and the main cellular structure. Anyways, eukaryotic genomes are huge. Yes, it is enormous. Again, size of the cell is going to matter. In order to store a lot of that genetic information, we need a bigger cell. Hence why a lot of eukaryotic cells are 5 times, 100 times bigger than bacterial cells. Bacterial cells are very simple type of organisms, but with eukaryotic cells, with the amount of regulation, with the amount of genetic variation, it's going to be huge. And we can see right here, based on the bottom of the graph right here, you can see the amount of nucleotide pairs per haploid genome. So half of that genome, again, one set coming from mom, one set coming from dad if you're looking at a diploid organism. But haploid genome means that it's just the unique set chromosomes. Okay. In approximation size right here, we have 4 times 10 to the 6 or 5 times 10 to the 6 base pairs long, nucleotide pairs long for E. coli. So that's a bacterium cell. And if we just go to humans right here, again, E. coli only has 4 times 10 to the 6. So approximately 4,300 protein coding genomes versus humans, we have 3,100 times 10 to the 6, which gives us roughly around 20,000 different types of protein coding genes. Okay, so it is a vast, vast difference. We have yeast cell, Saccharomyces cerevisiae, which is about 3 times bigger than the E. coli DNA strand right here. Okay. We have Arabidopsis thaliana, which is a plant that we use quite often. We'll talk more about that in the next section when we're looking at model organisms, but this is a very, very nice model organism. So a lot of these right here are actually introducing you guys to the model organisms. We have the roundworm, we have the Arabidopsis, we have the Drosophila, we have the zebrafish, as well as the mouse and the Homo sapiens. So these are all modeled organisms that we are going to be discussing right now in the next section of this chapter. Okay, so eukaryotic genomes are rich in regulatory protein, and again, they're defined. The genomes define the program of multicellular development, obviously. Okay, and so we're not going to talk too heavily about that. You guys get the idea. But now we are in our last section of the chapter. And I'm hoping that this has been a very good, just touch up review. Again, we're not going super in detail. I am repeating myself for a reason. But now we are going to take a look at modeled organisms. Now, model organisms, why are they such a good tool to use in scientific research? Well, again, we can take a look at their genome and we can see what happens when we mutate certain genes and discover or kind of observe what the phenotype or the physical or genotype traits are through that mutation to better understand the cellular processes, to understand genetic modification, etc. Okay. Molecular biology began with a spotlight on a bacterium and its virus. The focus on E. coli as a model organism has accelerated many subsequent discoveries. Again, this is very, very important. It doesn't sound as important as it is in the field. It's really, really important that this actually happen. Yeast serves as a minimal model eukaryotic. Again, yeast cells are eukaryotic organisms, so don't forget that. Expression levels of all genes of an organism can be determined, yes, through a lot of various testing. And Arabidopsis has been chosen as a model plant. So if you guys ever get a chance to take a plant biology course, or a herbology course, you guys are going to be looking at Arabidopsis a lot. I did get to take it with one of the instructors at the university who was I believe she was the what do you call it? The consult for the Avatar film, for the James Cameron Avatar film. So, she was a huge proponent for her teaching the actors how to correctly use scientific tools on screen. So that was really cool. I got to take a class with her in a plant biology course. So, anyways, don't need to know that, but just thought I'd share. Anyway, so model organism. The world of animal cells is mainly represented by a worm, a fly, a fish, a mouse, and a human. For insects or for other studies in the fruit fly, Drosophila provides a key to vertebrate development. Again, understanding how development occurs through protein concentration, expression, etc. The frog and the zebrafish provide highly accessible types of different vertebrate models. So we can see the effects that happens on zebrafish if we mutate a certain gene. There were a lot of genetic labs that I had a chance to join in that dealt with a lot of the zebrafish. And so if you guys are able to join a lab that works on zebrafish, that's awesome. The mouse is a predominant mammalian model organism. If you guys are planning to go into research and you're going to deal with mouse cells or mouse organism, you guys will need to undergo a lot of training and clearance before working with them, just because they are a more complex organism. And COVID-19 pandemic has focused scientists on the coronavirus, which is a viral study. Okay. So humans are unique in reporting on their own peculiarities, and to understand cells and organisms will require mathematics, computers, and quantitative information. Now, with the introduction of artificial intelligence, we're able to use a lot of these theoretical models to kind of figure out what could potentially happen prior to doing clinical trials in the industry. And so this is just a I wouldn't say a pioneer, but this is a huge leap forward in really discovering, different types of protein structures, how they fit within the cellular process, and determining what structures is needed to really fit into those active sites on receptors and other proteins. And so, again, this is just an introduction to those ideas. So mutation reveals the function of a genome. So we can see right here, here are two organisms. I believe these are just yeast cells right here. But in the first image right here on the left side, we can see this is the wild type or the normal type genome. So this is what occurs naturally, hence why we call this wild type. Now, for a test that we could do, we could insert some sort of genetic material into the DNA of this yeast cell and really see what's occurring. Now, when you're dealing with yeast cells, you're going to create a couple colonies. So you're going to create a colony, and then from that colony, you're going to separate that so that it develops its own colony, but it's essentially an identical model genetic wise. And so with the second colony, you're able to inject some sort of DNA or delete certain DNAs using various scientific methods. And we can see that with the injection or deletion of certain genetic material, we can see that the cellular structure of the second E cell has been mutated or has formed abnormalities or developed extracellular structure. And so we can see, phenotypically, we can see that we have this mutated yeast cell versus the original yeast cell right here, or bacterial cell. I think this might be more of a bacterial cell than a yeast cell. But we can see the difference physically just through this process alone. So mutations are a wonderful thing when you're working with bacterial cells or simple organisms, and you can see the effects of deleting or inserting different types of DNA. Okay. Molecular biology began with a spotlight on Y bacterium and its virus. Again, this is a very, very famous structure where we do see the virus shape. You have the shell that holds the viral DNA right here. So the viral DNA will be found in here. And then you have the landing mechanism and the insertion mechanism down here. I forgot off the top of my head. I haven't taken virology in a super long time, but this is one of the famous structures. Can't recall off the top of my head, but it is pretty much showing how a virus will inject the DNA into the cell itself, adding in and fusing in with the bacterial cell DNA, causing the bacteria to produce more and more of that virus shell-like structure. And the shell-like structure is usually a glycoprotein structure or a sugar shell structure that would interact with the components within a cell. Okay, so a focus on E. coli as a model organism has accelerated many subsequent discovery. Yes, again, E. coli is one of the biggest organisms that we do work with. And we can see the origin of replication right here. We can see the genetic material all the way around. And it's about 4 billion nucleotides pairs long. Okay, so we've fully dissected the DNA of the E. coli cell, and we can pinpoint different types of areas where we can study the effects if we mutated or deleted that variation. Okay. Now, your book is going to go more into the specifics of why these are, so I recommend reading it through. But this PowerPoint has gone for quite a while now, and I'm trying to condense the main ideas of it. But you will be required to kind of just brush up on the readings. And if your textbook does go into something a little bit more detail, it is a good idea to kind of read up on that. But I'm hoping that this lecture gives you that foundational structure before you go into the super nitty-gritty detail. Now, again, I will be going over the main things that I would like you guys to know. But for those extra details, please refer to your textbook. Anyways, yeast serves as a minimal model eukaryotic model. The reason why is because, again, yeast cells are incredibly simple. They are very simple. They're not as complex as a multicellular organism, but we can start mutating them or injecting them with different types of medicine or different types of viruses to see the effects on a cellular level. Okay. So again, here is an example of a yeast cell right here. And we can see how simple they are. They have a cell wall, they have a Golgi stack, Golgi apparatus. We do see the nucleus, and we do see that mitochondria, that archaic mitochondria, as well as a lot of ribosomes throughout. Okay, so again, scaling 10 microns to 1 micron. So budding yeast cells, again, it is a diploid organism, so they will have two sets, one from a parent and one from the other. Or if it does a symbiotic relationship where it splits on its own, then it will just mutate, or not mutate, but it will undergo cellular division through mitosis. Okay. But for us, for a yeast cell, it is going to be undergoing meiosis, okay, and sporulation. So there will be some spores that will be released. It's triggered through starvation. We can see that as it proliferates, it's going to go through spores, and then with the spores, it's going to land on an organism and then start hatching, or land on a surface and then start proliferating through there. So you can either have a proliferation of a haploid cell or a diploid cell, it doesn't matter. But once it has a haploid cell, it will combine with another haploid cell to form a diploid organism again. And so that's just how it's going to replicate. So depending on the certain process of its life cycle, whether it's undergoing the genetic variant cell division, or if it's undergoing its own identical cellular division, uh, it will be determined from there. Okay, mating usually immediately after the spores hatch. Again, the spores have to hatch first before two haploid cells come together, forming a fertilized cell. Okay. Expression levels of all the genes of an organism can be determined. Arabidopsis, again, has been chosen as a model plant. So here's an imagery of a Arabidopsis. It is a very, very small plant. It's a couple centimeters long. You can see right here, it's 1 centimeter. It can grow up to maybe 2 or 3 inches, and that's it. But it's a quick growing plant. It is a very simple plant, but we have the image right here, and we can start playing around with different types of mutations, especially if we want to inject certain proteins and we want to see where the proteins are falling in within the cellular structure. We can attach them with some fluorescent proteins with the modeled protein that we want, and then we can see it cellularly going through the cells here. Okay, again, I'm introducing a lot of different just uh, uh, cellular techniques right now. Don't need to know that in chapter 1. The world of the animal cell is mainly represented by a worm, a fly, a fish, a mouse, or a human. Again, we do a lot of studies based on these modeled organisms. With worms, there are certain studies that are being done right now where we're doing electrical stimulation and seeing the effects on healing through electrical exposure. So this deals within the idea of regeneration process. A fly, again, is understanding the development process. A fish is slightly more complex than a fly and a worm, and a mouse is one prior to or one after that. And so, again, here is a regular roundworm. The wonderful thing about a roundworm is that you can cut a roundworm in half. Depending on the species, you can cut them in half and they will regenerate on both sides. So it's a very, very great model organism currently in the regenerative field of medicine or of science. Drosophila right here. We used to do this in your 212 labs. One of the labs towards the end was for you guys to grow Drosophila flies, but we've had a lot of issues growing them in the past. But it would have been something that you guys would have done in your 212. And you would have been able to see the mutations because we would have exposed them to different types of mutations. But it was a little bit more complicated to run those labs. So, but for our case right here, you can see a wild type Drosophila fly. This is just a regular fruit fly. Okay, so this is your wild type, and here is your mutant. Okay, so here's your mutant, and we can see that the antenna grew into legs here. It's just the deletion of a certain genome. It could be an introduction of a new protein that cause a mismatch during the developmental stages of the fly's embryonic development. Okay. A frog and zebrafish provides highly accessible vertebrate models. Again, these are more complex organisms, and if we want to go from model organism trials to clinical trials, we have to pass certain stages, especially in the biotech field. Okay, so we can see here is your fertilized egg, your zygote in hour 1. We can see the morula right here developing. So morula, okay, is just a ball of 10 to 32 cells. Once it hits into a blastula, which is 16 to 32 cells, so morula is right before the blastula. Once it hits the blastula stage, that's where we're going to start forming the inside works, where a cavity is going to start forming inside here and then start developing the foundational layers through gastrulation, neurulation, or organogenesis. So gastrulation or gastrula is the next stage where we start forming the ectoderm, endoderm, mesoderm. Neurulation is where we start forming the notochords and the spinal cord, as well as the nervous system. And then organogenesis is the development during the system, organ systems, where we start laying everything and connecting everything together. And then tadpole is the next bit. Now, I went over a little bit more of the embryonic development. Guys don't need to know that, but it's just a good idea to kind of refresh your memory. I believe you guys have done developmental animal development within your Campbell textbook. So it's just a review. Okay, so here's the zebrafish. You guys can see it. We grow these quite often. They're great models, and you can actually see the fluorescent proteins right here, the red, yellow, and green fluorescent proteins that we've injected into the DNA, or not to the DNA, but into the organism itself to see where these imageries or where these specific things that we're trying to follow are. Okay. I don't think they're translucent. I think these are actual GFPs and RFPs and YFPs. Okay. Mouse is a predominant mammalian organism. Again, these are laboratory mice. We genetically grow them. Not genetically grow them, but we grow them to be genetically identical to each other. And it's a great modeled organism. And so right here is again just the percentage of amino acids identical in the hemoglobin chain, so in the blood chain itself. So we can see, from 0 to 50 to 100, we can see how many amino acids are identical based on the blood. Okay. Over however many millions of years. So we can see that for humans and lizard to be the same, we have 57% of the hemoglobin alpha chain identical to each other. Humans and lampreys, I believe these are eels, are 35. Humans and tunas are 55. Humans and chickens are 70% identical. Humans and chimps are nearly identical. It's 100% about. Okay. So again, just mammalian babies right here. And COVID-19 has focused scientists on the SARS CoV-2 coronavirus. And it's a virus. And so there's a lot of glycoproteins found on the shell of the virus, and for them to be injected into a eukaryotic host, it needs to be able to be received very easily. So with glycoproteins on the surface, once it's injected into the cell or once it's introduced into the cell, then it's going to release its RNA, fusing with the host DNA, and then from there, uh, uh, proliferating. Okay. Again, we require math. We have a lot of models nowadays that helps with the understanding of different types of DNA, RNA, protein coding regions, etc. Okay. And that's pretty much it. So I'm hoping that was a great introduction to chapter one for you guys. There's a lot of stuff that we touched on, and I tried to introduce stuff from past lecture past courses for you guys. And so there will be problem sets that I will be introducing during class. But for you guys, if you guys want to know, look at the end of your chapter. There's a great number of problem sets at the end of the chapter. I recommend doing the odd ones because I believe your textbook gives you the odd answers, but I recommend doing at least 2 or 3 of them per section. So there's 4 sections here. Do 2 or 3 per section so you have a good understanding of it. You might need to refer back to the textbook for more details on specific reasons why we choose certain model organisms. Again, I'm telling you the brief reason, but hoping that these lectures are going to be giving you guys the edge so that when you come to class, you have questions ready to go and we're able to answer it together as a class. And so that's it. That's all I have for today. Good work finishing the first chapter. Next chapter, we will be talking about the properties of atoms again. So this is very similar organization to your prior textbook. We're going to be talking more about the macromolecules, the water, the behavior of water, atoms, chemical behavior, atoms, bonds, and everything. So pay attention to that. It's going to be a nice review of your GChem, OChem material. And then we're going to get started into the biology once we hit chapter 3. All right, anyways, guys, that's it. Good luck and I'll see you guys in the next video.